Hello, I'm Mary Ito, and welcome to CRAM. Thanks for joining me as we explore new and innovative research and big ideas that may change the way you think and act. Now, were you a fan of the popular TV series CSI, Crime Scene Investigation? Or maybe you were a fan of one of its countless spin-offs. If you like nothing better than tackling unsolved cases, you are in good company. Amro Zayed is a professor of biology at York University in Toronto, and he is heading a huge research project. It's called BeeCSI, and that's B as in bumblebee. What is killing them? And what can we do about it? For many years now, researchers have been scratching their heads over declining bee populations. And now Amro and his team are sleuthing their way to a better understanding of bees and how to ensure they survive and thrive. Hi, Amro.
Hi, how's it going, Mary?
It's going great. And how about you? I bet you're busy as a bee.
I'm doing well. Of course, yeah, just buzzing from one Zoom meeting to another.
Okay. Well, listen, I wanted to start off and talk to you about the importance of bees. Tell us how vital they are to us and the planet.
Yeah, bees are extremely important to us. They pollinate about a third of every mouthful of food we consume comes from bee pollination. And it's important to note that there are actually many species of bees. There are about 20,000 species of bees worldwide. And when we think of bees, we think of honeybees, we think of bumblebees, but there's thousands of other native bees, and they're all super important for pollination. And obviously, from our perspective, in agricultural settings, bees are important because they pollinate crops. A lot of the fruit we consume in Canada comes from bee-pollinated crops, and things that you don't normally think of, like canola, right? So bees are important for canola pollination, and obviously the oil products that come from canola. But there's also just all of the native bees that are out there in our meadows and ensuring their reproductive potential and reproductive output of our native plants. So they're really important for agriculture, but also the world would look very different if bees did not exist and plants did not rely on bees visiting flowers and moving pollen from one plant to another.
I can't get over it. You said they're responsible for 1/3 of the food we eat. And I did read that somewhere. It was like 1 out of every 3 bites we take, a bee is responsible for that. That's incredible.
And it is incredible from kind of we need them. Humans need to eat to survive, and we consume a lot of fruits and vegetables which have a lot of nutrients that you wouldn't necessarily get if you were to go on an all grain diet. And if you could put a dollar value on it, and in Canada it's about $5.5 billion per year. That's the amount of our productivity, agricultural productivity that comes from bees visiting plants.
Tell us about some of those amazing facts about bees. They kind of remind me of our neighbors. We see them all the time, right? And yet, how well do we know them, right? We see them every season. They come in, then they leave. Tell us about some of those curious facts that you've come across over the years.
Yeah, so there's a lot of bees that are specialists that would only visit a certain flower. So for my PhD work, I spend a lot of time sneaking into people's gardens early in the morning, not like backyard, but front gardens. I would kind of walk in people's driveways and look for sundrops, as evening primrose, lovely bright yellow flowers. And if you're around Toronto and you wake up early enough, you'll see this small, tiny little black bee that visits the blossoms, and the small little black bee only visits and only survives on evening primrose pollen. So
what's it called?
A lot of people don't realize.
What is that?
It's called they changed the names a lot. I think the latest name is Lasioglossum enotheri. It's a sweat bee in the genus Lasioglossum, which is in the family Halictid, normally called sweat bees.
Okay, you're losing me there.
They would actually land at you. Yeah, they're called sweat bees because they would also land on you and like lap up some of your sweat because they use the salt.
Oh, that's handy. Okay, this is weird. You
Yeah,
said
yeah.
that the names keep changing. Why would names keep changing?
So it's something actually that my former supervisor and now colleague Lawrence Becker works on a lot. But yes, you have a name and then you study more and you realize that some of the relationships that you thought kind of existed were not there, so you have to revise the name. In which case. For that bee, people thought that it was different enough that it warrants its own genus name. But then they realized, I'm like, no, actually, it's not that different, and let's just call all of the bees with these kind of characteristics one genus called Lasioglossum. Yeah.
Oh, I see.
So
I had no idea that names keep.
it changes
Yeah.
quite often. Yeah,
Wow.
the species name doesn't change, but the
Okay.
genus name could be. Some people like to kind of split things and have their desk into small little compartments, and some
Yeah.
people like to merge things and say, well, all of these little bees are pretty much very similar evolutionary, so we're going to lump them together as one genus.
Gee, I'd hate to be in charge of the family bee tree because that's confusing if it keeps changing.
It's fun. It's fun. Yeah. I actually have a bee named after me and my my
Really?
wife.
I didn't know that.
Yeah, my wife's first name is Jen, and I'm Amro. So there's this bee called Jen Amro as this species name. We actually were the first to collect it while we're doing field work in the deserts of Chile.
Is now would that be
this small little bee that you would never ever like notice or very few people actually collect it, but we're very happy that
that's amazing. Is it in Canada?
Got named after us.
Is it here?
No, it's actually found in Chile around the foothills of the Atacama Desert.
Oh wow, that's well. There's your claim to fame.
That's right.
Well, actually, no. I should say you've got more than that one claim to fame because you made a discovery a number of years ago. You found out about a clean gene in bees. Tell us about that.
Yeah, it was a very cool project, and I can I give a shout out? I'll give a shout out to my PhD student Brock Harper, who's now a professor at Purdue in Indiana. But him and I and several others in Canada were working on this really old problem in honeybee biology. So honeybees are social. They live at these amazingly complex colonies, up to 80,000 individuals, and when you're social, sometimes you evolve kind of behavioral traits that a single individual wouldn't necessarily make use of. So for humans, we have a lot of hygienic behaviors that prevent disease transmission, right? So putting a mask on, washing your hands, we know these are things that could limit transmission of COVID. Bees, because they are social insects and they live in these dense environments, they've also evolved a lot of hygienic behaviors. And one behavior in particular that's been known since the 60s is the ability for bees to kind of detect dying brood. So a queen lays its egg in the wax comb, and the egg hatches into a little larva, and then eventually that larva kind of the workers put a wax to cap the cell, and the larva goes through a process of metamorphosis. But sometimes during this process, the bee gets sick with a virus or something, or gets infected by a parasite. So bees have the ability to kind of sniff out these dead bees inside the wax cells, and what they do is they uncap the wax and remove the sick, dying larva, and they kind of throw it out of the colony as a way to kind of prevent the transmission of mites or pathogens within the actual beehive. So this behavior is known as hygienic behavior. And it's been known since the 60s, and there's a lot of kind of debates about, well, is it genetics? And of course, it has a very strong genetic component. And there's this race to find out the genes involved with this trait. So myself and my PhD student and several colleagues across Canada were actually able to sequence the genomes of bees that exhibited this behavior, that were really hygienic, and bees that were not so hygienic. And by comparing these genomes, we identified about 70 genes that are essentially orchestrating, they're underlying this ability of bees to detect a dying brood and then perform that behavior of uncapping the cell, removing the dying brood and throwing them out of the colony.
Wow. So they're sort of the cleanup crew, aren't they?
They're exactly the cleanup crew. Yeah. And it's interesting in that it's not like it's not one bee that does all of these things. It's like a coordinated work. It's
teamwork.
very cool, right?
Teamwork.
So yeah, it is teamwork, right? Somebody has to kind of sniff out the dying bees, and then they uncap it, and then different bee goes in and removes the dead brood. It's pretty cool. It's pretty amazing. Yeah, honeybees are pretty amazing. The things that they're able to do is just kind of mind blowing.
Really? So what else about honeybees should we know?
Well, I mean, like the other, I think there was probably a Nobel Prize waiting for the person to actually get the genes that are responsible for the waggle dance, right? So the first Nobel Prize in bee biology, probably the only one, is the discovery of the dance language. So how honeybees are able to visit a profitable flower patch and come back to the colony and do a little dance that then lets their sisters know the location of where to forage and how far from the colony. So this is a language that's symbolized in a little dance. It looks like a figure 8 buzz that the bees do, and the direction is relative to the sun angle, and how far they buzz is the distance, how long they need to go. It's mind blowing.
That is totally mind blowing.
Yeah.
Okay.
So we know how.
Hold it. So
yeah, I was going to say we've known around this for a long time, and we are able to decipher the dance, but we're still not sure what the molecular machinery. Where is this dance? Where are the dance genes? That's what we're going to get. The genes for dancing.
But you've said scientists have actually been able to analyze and determine what every movement means.
Yeah. So you could spy on bees. You could put a little camera in, and looking at their dances, you could decipher where the bees are foraging.
So are you? So when you said it's like a figure 8 that they're using,
yeah,
right?
Yeah.
Can you actually translate that information of the dance to actual distance? So you look at it and you say, oh, this is how far that flower is where they should be going to pollinate, or you can actually make that exact accurate interpretation.
Yeah, I mean it'd be there'll be plus or minus, but the angle is pretty clear. Like when you know when the dances are occurring and where the sun is, it's really easy to translate that to a vector. The distance is a little bit relative, but you could essentially calibrate it to the other dances in that day, and you could get a sense of the general location of where a bee needs to fly to find this nice batch of canola or echinacea or sunflowers.
That is amazing. Doesn't it make you wonder, well, why would it be just limited to that? Maybe there's other kinds of language going on that you just don't realize in their movements.
Yeah, and we know that they have one of my favorite behavior is the motivational buzzing. So how is this, Mary, for something fun? You're working, let's say in a studio, and let's just say somebody's not working as hard as you wish, and you could just go up to them and you shake them and vibrate them. That sounds pretty wild, but it bees that this actually happens where you could have some workers just like buzz other workers, and it's perceived. I mean, we can never really get into the bees' brains and kind of understand why they do this, but it appears to be kind of a motivational stimulus. So
really, so and you're saying that bee,
we have work to do. We have work to do.
So that bee was showing sort of lazy behavior beforehand, and then when this other bee goes and buzzes, they actually just start working harder. They pay attention.
it's associated with, it's believed to involve that, some kind of motivation to either switch tasks or to perform a specific task. But the bees do like many cool things, and obviously
Yeah.
we're trying to get under the skin and trying to kind of understand what does it all mean.
Yeah, that's fascinating. And when I think about it, the fact that, these bees get after bees that aren't working as hard, it does make sense, doesn't it? Because it's teamwork and they live so closely together and they rely on each other so much that I guess, everyone's welfare depends on each bee working hard and cooperating.
Yeah, they're all essentially rowing on this boat, and the boat has to stay afloat and not sink, and they do need. There's still a surprising amount of, like, other people have studied this in ants, and there are cheaters. There are, like,
Just like
many
humans.
workers that. Yeah. So it's these societies are always kind of at the edge between kind of cooperation and conflict. So they do seem kind of harmonious to the outside, but not everything is as it seems to be. And there are social parasites, so to speak, in terms of not doing an unequal share of load, and there's policing in other contexts, and some of these workers could actually, like, activate their ovaries and lay unfertilized eggs. So there's policing from that sense, and in that, I'm working here. How dare you, like, lay eggs? That's the queen's job. So there's interesting kind of worker policing that happens within colonies as well.
Yeah, that's just like brazen behavior, isn't it? Laying eggs when it's the queen's job. Huh.
That's what the queen is there for, right? That's her kind of her only duty in the colony, is just to pump out the eggs.
That sounds like treason. But anyway, so, I mean, in spite of all these amazing behaviors that bees have to survive, essentially, right? I guess it's all about survival. That they're not. I mean, they're in trouble because we've been hearing about the decimation of bee populations for quite a few years now. So why is it still such a puzzle? I mean, why is it so hard to figure out exactly what's happening?
Yeah, it's a big problem because the history of this is that we've known since maybe the early in the century, maybe 1990s, early 2000s. There was reports of bee population declines, and I'll mention that. Obviously, when we talk about bee declines, it's a little bit unclear about which bee we're talking about. But there is a very clear trend in many native bees, solitary bees that nobody would ever kind of consider, but social bees like bumblebees and so on. We know that they've experienced population declines, and actually some bee species have gone extinct in some areas. And honeybees have also experienced declines in their health. It hasn't really led to precipitous declines in the number of colonies, so honeybees are not going extinct, but they've been becoming more difficult to manage because there's just a plethora of things that have come together to reduce their fitness. So the issue is that we don't really have good tools to be able to know what bees experience. So the situation is we have a bee population that was common, but it's no longer common. And well, where do we start? We could look at the environment, and we could say, okay, well, what's in the environment that could be potentially stressing out these bees? Is it agrochemicals? Is it pesticides? Are there pathogens? So typically, you would notice a decline, and then you would say, okay, well, there's all the possibilities, all the things that we could think of, and then you go through and systematically test them, kind of one by one. And that type of research takes a very, very long time. And it essentially it requires you to come up with an idea of why the bee is declining and test it. So you only really test for the things that you could think of. So it leads to a bias in some way, right? I think pesticides are important. I'm going to go and test the bee, and I found pesticide A. Does that mean that A is causing the decline? It could be that B, C, and D are also involved, but I didn't think of them, so I never bothered to look. So it's a very slow process, and it's inherently biased because it requires us to kind of think about what are the stressors and go and test them. So that's where the bias comes in. So I've been thinking a lot with how to kind of improve this, and I think early about 10 years ago, I'm like, well, instead of going out and actually thinking and trying to come up with what's stressing the bees, well, why don't we just kind of like ask the bee, right? And obviously it's crazy, you can't talk to a bee, but we can look at the bee's internal physiology and internal systems and try to get a sense as to why what bees are being exposed to in the field. So looking at the bees themselves for the answers, and not just focusing on what could be in the environment.
Okay, so elaborate on this, because this is, I guess, at the crux of your research.
Yeah, so it's kind of like, how humans get diagnosed, right? You feel sick, you go to a doctor, they do a battery of tests on you, blood work, imaging, X-rays, and so on. They're just trying to get a look inside and get a sense of what's wrong, what's weird. We had this idea that we could do this. We're not going to go give bees MRIs and X-rays, but we could sample some of their tissues and look at the activities of genes that are getting turned on or getting turned up or down in specific tissues. And the idea here, at least to us, it sounded pretty simple, right? So, if a bee is getting sick with a pathogen, well, bees have an immune system, and we should be able to see signatures of a bee mounting an immune response, right? And a bee should be upregulating proteins and enzymes that are antimicrobial. If a bee has been poisoned by a pesticide, there's a whole set of other enzymes and systems that come in play. Things like detoxification enzymes, right? Bees have specific molecules that recognize any foreign chemicals like pesticides or anything nasty produced by plants, and kind of binding it up and chopping into little pieces and secreting it up. On the internal physiology of the bee, we could then use that to guess the actual stressors that are influencing the fitness of a colony or a specific population.
Wow! So this is your BeeCSI research project, right? I mean, this sounds massive. You got $10 million to do this? That sounds like a lot of money.
We are very fortunate to receive this grant from Genome Canada. I think it's probably like the largest grant for bee health. And it's a very large team. It's not just me at York University. We have colleagues from University of British Columbia, all the way to University of Laval, and folks from the government, so Agriculture and Agri-Food Canada. So it's a very large team, and it's a very ambitious project. So the idea here is we're going to take the honeybee, and we're essentially going to take honeybees and throw the kitchen sink at them. We're going to stress them out with every single thing that we suspect or can think of that might lead to decline in their fitness. And then every time we do one of these experiments, we measure the activity of genes and proteins and actually changes in their gut microbiome. And through this process of getting thousands, about 2,000 samples of bees stressed out from different combinations of things, we're building a database where we say, okay, every time a bee has been exposed to this pesticide, I see gene X being upregulated. And every time I see a bee getting exposed with this mite, then I see gene B get change in a specific way. So we're developing this database that will then allow us to, instead of actually looking for the stressors themselves, is looking at the internal state of the bee, which genes are going turned up, which genes are being turned down, which gut microbiome, which species in the gut microbiome are shifting, and then use that to render a diagnosis to say, okay, well, based on the pattern of gene activity, your bee's been sick because it got exposed to neonicotinoids, it was malnutrition, and it was also sick with the virus. So take the internal state of a bee and give us a specific diagnosis that then beekeepers could do something with.
Wow. And are you doing all this in a lab? Like, where are these 2,000 bees you're getting?
So it's actually probably like 2,000 groups of bees, like either colonies or several bees in cages, and they're in colonies. So the bees start out being in colonies. So we have places in Quebec, a couple of places in Ontario, Manitoba, several places in Alberta, and several places in British Columbia where we're keeping these colonies, and we're essentially taking them either in the lab and doing the experiments in the lab, or doing the actual experiments in the field. And some of the experiments involve, putting bees in gradients of stress. So, a colony in a nice pristine national park or near a national park to a colony, right near a canola crop or a blueberry crop or a cranberry. So we're putting bees in places where we kind of expect them to be exposed to different types of stressors in terms of the amount of agrochemicals they're getting exposed to, or the nutritional, the amount of nutrition that a given environment can provide.
Yeah, I mean, I don't know if it's much like humans, but I would think that doing an experiment in a lab, the results you get could be quite different from in its natural setting. Right? Because people don't live in labs and bees don't live in labs,
exactly.
Right? Yeah. And there would be so many. But again, if you put it in the natural setting, now there's only so much you can do to control the environment. There must be so many factors that you have to account for.
Mary, you nailed the problem on the head. Is that the lab gives us like cause and effect. We have full control. We can experiment things. In the field, we don't have cause and effect, so we have kind of correlations. We see things that might change together, but is it because one causes the other? We don't know. So the solution to this is actually you do both. So half of our experiments are done in the lab where we could get cause and effect, and then we go to the natural world and we see whether the knowledge that we've gained from our cause and effect experiments in the lab, whether they translate. The stressor, the biomarkers, we're calling them biomarkers. That the idea that a gene, that if we know that how it's being regulated, could mark what stressor the bee is exposed to, that we're calling that a biomarker. So the biomarkers we identify in the lab, we're going to test whether they actually are still predictive in the real world. So if I see a colony that is turning on gene A, and I say, okay, well, gene A should only be turned on if I am exposed to a pesticide. Is this colony now in the wild, is it being exposed to that pesticide? And we know because we look for 200 different pesticides, and we're like, yeah, okay, it's been exposed to the pesticide. So the biomarker is now validated, and we could use this knowledge that whenever we see this gene, we know it's this specific pesticide that the bees have been exposed to.
Wow. Very CSI, right? As you go through all the different factors and eliminate one after another.
Exactly. Yeah. The name, I'd like to take credit for the name, but I actually, when I started thinking about this, I called it Bee Doctor because well, I thought it was clever because, well, we're trying to play doctor with our bees, and I have a PhD, so I'm going to call myself a doctor.
The bee doctor,
and then I was pitching it to some colleagues at Ontario Genomics, and one colleague is like, you should call it BeeCSI, and I'm like, oh my god, yeah, that's like. That's it. That's the name.
It's a great name. And how far along are you right now with research?
It's a 4-year project, and we are, I think, in it just started year 3 now. Things have been, with COVID, it's tough doing research with COVID because there was like a 6-month period, but we're trying to kind of catch up, and things are on track.
I get you can't disclose anything yet, can you?
We're still in kind of the data acquisition. So we just got this huge, forgot how many terabytes of data, and we're like, wow, we have data now. Because with these projects that require field work, you kind of spend the first half of the project just concentrating on doing the experiments, and then the second half is when the fun doing the experiments is fun, but when you actually get the fruits of your hard work, and you get the data, you get the genomes, and you start looking for markers and so on.
Oh, wow. So one more year to go. We'll definitely be keeping an eye on that. I mean, tell me, you've got so many researchers involved. I'm sure there are other people, bee enthusiasts, bee conservationists. They're watching your work. How much of a game changer could this be as far as, sort of detecting what's at the root of this problem of survival and helping them, survive and thrive?
Yeah, I think if the experiment works in how we kind of anticipate it will work, I think this will be a total game changer. Because like right now, it's just that technology doesn't exist. Like people think that beekeeping is a highly technical and people think it's like a type of farming and like livestock management and there's a lot of tools and so on, but it's actually like very, very difficult to diagnose things. I mean, I get personal calls from beekeepers. I've observed X, Y, and Z. What do I do? And it's very difficult. And often, I think the real problem is that the only time when beekeepers test for things is when colonies have died and they want to know why. So it's kind of like too late. Right? If the colony's already dead, they're just, that's it. You've incurred the loss. What we're hoping to do is have these tools come in, so that way you know what's happening in your colony and you're able to do something about it before it's too late. So this could potentially be transformative from the industry, because it could prevent a substantive number of colonies from perishing. Part of the problem of having so many colonies die every year is that we as beekeepers rely on importing queens from outside of Canada. And actually, this almost led to a catastrophe last year with COVID. I like to say that, but our team actually has a COVID paper out, and it's a paper on how COVID influenced the beekeeping industry. And there was all of these restrictions on importing queens and labor and so on, and it was a very close call. So by
Sorry,
reducing,
can I just ask you? Yeah, I didn't even know that queens were being imported. Like, are you importing it because we don't have enough queens? What's going on?
Yeah, so because when colonies die, typically they die in the winter, and the mortality rate could be substantial, right? Like, so on average, about 25% of the colonies die every winter. Sometimes it goes up to like 40%, and so you go early in April, and okay, well, you have 100 boxes, but only 60 of them have queens, so you need 40 queens. Where are you going to get those queens? It's too early in April for Canadian beekeepers to actually produce their own queens, so they have to import them from outside. And the numbers are amazing. We're talking about anywhere between 200,000 to 300,000 imported queens every year.
Wow. And so these colonies that are dying over the winter, that's not a natural death. They're not dying every winter just because it's the winter. They're dying for other reasons.
So we know what natural death from just like having a cold winter is, and historically, like the overwinter mortality is somewhere between like 10-15%. So the overwintering mortality rate has been substantially elevated from the historical records, and things that essentially affect the health of the colony. So you could have viruses, or you could have pesticide exposure, and it might not kill the colony in the fall, but it weakens the colony such that when the really harsh winter comes in, that kind of that it's a threshold kind of effect where, okay, if you're just not strong enough, you're not going to get through the winter.
Right. Wow. So we're really up as far as the percentage of colony deaths then over winter. Yeah.
Yeah. And what we're hoping is that these tools could essentially, if used, and then if we are able to give the data back to the beekeepers within a short period of time, that they could actually like, okay, yeah, this colony needs to be treated for X, Y, and Z. Then they'll do the treatments. The health of the colony will increase, and then they'll be able to survive through the winter.
Yeah. Is this kind of a race against time? I mean, do we know how long we have already? I mean, as you've said, well, bees are in trouble. I mean, Is there kind of this, not a date, but some sort of idea of range of time when, oh my goodness, if we don't get to this problem in this way, we are really in serious trouble with the bee populations? Do we know anything about that?
Yeah, I think for honeybees, certainly there is a lot of urgency. We are trying to work as fast as we can to get these tools out for the honeybee market. But I would say that the race against time is actually more true for things like native bees, bumblebees, where extinction is forever, right? There is enough honeybees that yes, locally we might be short on queens one season,
right?
But it's more like an economic problems for beekeepers as opposed to this actual species going extinct.
Okay.
But for bumblebees and native bees, yes, there is a definite urgency, and we're hoping that that these kinds of experiments and these BeeCSI type way of thinking about diagnosing bee health could be applied to declining bumblebees. So actually, we just published a study with my colleague at York University, Professor Sheila Kola, and she's an expert on bumblebees and declining bumblebees. And there's this yellow-banded bumblebee that's been declining in Canada. And again, why is it declining? And there, we have several long lists, but it's difficult to kind of put a specific stressor. So we applied this kind of same genomic technology. We took workers from Ontario and Quebec, and we're able to measure the genes that they're turning on and off in their tissues. And we show some pretty clear signatures of exposure to a couple of new classes of pesticides, as well as exposure to pathogens. And actually, by sequencing, we're able to actually sequence the pathogens themselves. So we're able to say, yeah, we have a signature of the bee mounting an immune response against pathogens. And then we actually found the pathogens themselves. So it was very transformative in that, going from Bombus turrica is declining, and it could be because of climate change, pesticides, pathogen, poor nutrition, to it's yes, it is pathogen, but now we know it's like X, Y, and Z. And in terms of pesticide, there's two chemicals that we implicated that might be at play. So it goes from kind of just very nebulous general knowledge that might not be actionable to a more specific knowledge that could be actionable in terms of planning how to recover and how to manage the species, so that way we reverse the declines and going to a more kind of stable and increasing their population numbers.
Right. Well, yes, I see that approach of managing and helping the populations. But what about the responsibility on the other side? If you've narrowed down what the pathogens are and you actually know the chemicals, right? Then does the responsibility also fall on we as humans to do something about those pesticides?
Yeah, and obviously it's the bee health is a complicated area, because a lot of the stressors are extrinsic, and obviously the stressors that are associated with agriculture, they're it's not like these pesticides are just naturally found in the environment. They're being used to control pests in some other crops. So this knowledge then leads to kind of a more informed conversation between regulators and people interested about saving bees and farmers, and how we could have sustainable food production without endangering our bees, which are key for food production, right? So I think it's not. I don't want to trivialize the problem, but at the end of the day, I think once we kind of implicate the stressors, I feel like farmers are fairly reasonable, and they realize that the importance of bees in terms of crop production. So I think once you know what the issue is, and you implicate compounds, and there's a lot of potential for having good discussions and with regulators and the farmers, and either restricting the use or just changing the use in a way so that way it doesn't impact bees. a lot of the issues that we had before about controversies with pesticides and bee health, a lot of it was just based on the uncertainty of like, well, are neonicotinoids really like hurting bees? Are bees really exposed to substantial level of neonicotinoids? These conversations, without having diagnostic tools, they could continue forever, right? No, bees are not exposed. Yes, bees are exposed. We'll prove it, and then you spend like years. With these tools, I think that well, are bees exposed? Here's a test. Yeah, okay, they're exposed. So let's move forward and then talk about how we're going to mitigate this exposure risk. So we spend less time arguing about what's out there and more time talking about, well, we know what's out there, and let's now move to the more productive step, is how we're gonna protect bees and how we're gonna manage our use of pesticides in a way that doesn't reduce the fitness of native bees and manage pollinators like honeybees.
I mean, as serious as the situation is with bees, are you encouraged by because it seems to me, just from being on the outside, that there has been much more interest, not just from researchers like yourself, but even from the public, to help save the bees, to do something about it. How do you feel about it?
Yeah, I'm really hopeful. So when I started my career in research as a graduate student, you couldn't convince the media to publish a story about bees, right? There was this, bees, they sting.
Well, they're annoying.
There's just a lot of
pest.
Misinformation.
Sure.
Yeah. There was just a lot of misinformation and lack of kind of appreciation about what bees provide, what bees bring to the table. They bring fruit and oil to the table.
That's right.
So that has changed a lot. Excuse me. And I think that's been great in terms of getting public support, because ultimately at the end of the day, we need public support for laws, for lawmakers to kind of make the world a better place for bees, and I feel like we've secured that. So now it's just a matter of like getting to the actual evidence and identifying the things that we need to kind of limit to help bee populations recover.
Yeah, that's so true. And I'm not sure. Well, again, maybe it's multifactorial as far as when the public's sympathies, change towards bees, but they certainly seem to, because you're right. Bees were thought of as being not annoying, just annoying, but, scary. You could get stung. Oh my goodness. Yeah. We people could die if they're allergic from a bee sting. The same could be said of, not to rain on the bees parade, but the same could be said for wasps, right? Because wasp populations are declining as well, but we're not hearing a ton about that. And all I hear is usually, again, it's like the old bee image. Oh, wasps, I hate wasps. They're so annoying. They're always in my food outside. I think they need an image makeover too. Don't you think so, Amro?
Wasps need to hire a good PR firm.
Maybe that could be your next project after you help save the bees, help save those wasps.
As a homeowner, I think a lot of my frustrations about flying insects are around essentially wasps trying to attack us and in barbecues. So I could see where the resentment towards like flying black and yellow things comes from, and I'm glad that the public perception about bees has really changed. Actually, I'm not sure if you remember, Mary, but I had my first ever interview with you back a long time ago. The Diploid Mare production. I was like so excited. I'm like, wow, Mary's gonna actually talk to us about like bee genetics. It's like, that's amazing. So I guess I'm trying to say you're a pioneer. Yeah, you're a pioneer in the public change in the public perception about bees. So thanks, Mary.
Well, listen, this is great, right? It's come full circle. Look how far you've come. Look how far bees have come, and hopefully, we're going to make more great strides, right, in the future, near future.
I'm very hopeful. Yeah, I'm optimistic.
Thank you so much for sharing your time and your knowledge with us today, Amro. Good luck with your work.
Thank you so much. My pleasure.
That's Amro Zayed. He is a professor at York University, and he is heading a team of researchers in a $10 million project. As you heard, it is called BeeCSI. What is killing our bees, and what can we do to help them not only survive, but thrive? We're going to keep an eye on Amro's research, and if you'd like to know more about it, you can visit our show page. Thanks for joining us today, and until next time, take care.